Good morning. I'm Chris Rebelo. I'm the Senior Director of Communications here at the Buck Institute. It is my extreme pleasure to welcome you to this month's Live Longer, Better Community Seminar. Today, we're going to talk about time. We're going to talk about why it matters and why you may want to become a Buck time traveler. I'll be joined shortly by Dr. James Yurkovich. This project is his baby. In the meantime, a few housekeeping notes. If you have not put your phone on silent, it would be great if you'd be willing to do that. And also, I always like to keep the fire marshal happy, and I point out the emergency exits at the back of the auditorium. A question: how many of you are new to the Buck? We have a few new folks, great. So rather than me going on and on about how exciting our science is, we have a short video that features the folks who are actually doing the work. Joran, make it happen.
The Buck Institute is interested in understanding aging, not only to make us live longer, but to actually alleviate and suppress the development of diseases.
Chronic disease is the great health challenge of our time. Aging is the main driver of chronic disease. To eliminate chronic disease, we must harness the biology of aging so we can live healthy longer. The Buck Institute is the world's first research organization dedicated solely to the biology of aging. We work with the building blocks of biology to understand how we age.
The Buck is concentrating on really trying to go to the root of the problem, so understanding that we can sort of intervene really early.
The Buck Institute is probably the best place in the world to be working on aging because there's so many different aspects of aging that are being studied. It's key because no one lab can do it all.
Our scientific explorers are brilliant, curious, and committed.
The idea that you could actually develop drugs that slow down the aging process was really something that 20 or 30 years ago people thought this cannot be done. Not only can it be done, but we will do it.
The pieces are in place. And we are in a unique position to put them together. Our world is defined by paradigm shifts, created by scientific discoveries. The biology of aging is the next frontier.
We're working toward a world where healthcare is better. It's a world where if we can eradicate the aging component to disease, we're all going to live healthier and longer.
It's just an incredible vision. It really will, surpass anything else that we've ever seen before.
We are inviting you to a future of aging without illness. It's happening today, and it's happening here. Join us.
I agree. We're going to get this. Okay, great. So before I introduce James, in your program, we have a list of our sponsors. I want to thank them for their support. If it makes sense to you, I want to encourage you to take advantage of their businesses if you can. That would be a great thing. I also want to make sure that all of you have cards. That's how we're going to do Q&A today. If you have a question, write it down, raise your hand. A volunteer will be by to pick up those cards and get them up to me. A special welcome to a bunch of folks who are joining us on Zoom today. You too have a chance to participate. Just enter your question in the Q&A section and somebody will get it up to me here on the stage. So It is now my pleasure to introduce Dr. James Yurkovich. He is an assistant research professor here at the Buck and is chief information officer at one of our partners, Phenome Health. You can read about James' educational background in the program. Suffice it to say, he epitomizes what it is to be interdisciplinary. He is helping us bridge the gap between technology and biology. I think of him as a bright star in the Buck firmament. We are glad to have him. Come on up, James. He designed this sign, so he's a cool guy. So, James, the first question, oh, I know. Can everybody hear me okay? Okay, if there's a problem, raise your hand. We'll deal with the sound. Okay, this is a little different for me. Usually I'm up here and I'm interviewing and talking to biologists or geneticists. Today I'm talking with an engineer, so that's a little different. So that's your primary training, James? What does an engineer bring to the party?
For me, it's all about bringing a different perspective. That's what's so valuable about interdisciplinary science and a lot of what happens here at Buck. I've made my career, like you said, kind of bridging the gap and translating a lot of the technologies. I have training in computer science, electrical engineering, bioengineering. Bringing all of that together, that's really what we need to solve these problems. No one person can do all of those things, so we need to be able to, bring people together to do them.
Do you think about disease differently?
Probably. I mean, I don't want to speak for all of my biologist colleagues, but I tend to, as an engineer, it's definitely a different way of thinking. It's kind of epitomized up here, but I'm very application driven. I think that's one of the things that sets me apart from maybe some scientists. I mean, I love doing basic science research, but I am wholly focused on applications. And so in thinking about disease, the end goal of a lot of what we do is to design effective therapeutics. And therapeutic might be a pharmaceutical intervention, it might be a lifestyle intervention, use that term kind of broadly. But if that's the end goal, then my perspective is to work backwards from there. What does it take to actually do that? And so if I want to be able to define an effective therapeutic, I have to be able to reliably track the system. Because if I intervene, I have to be able to see, did I actually make a difference or not? So we have to have robust metrics to do that. In the business world, we call them KPIs, key performance indicators. And in order to develop those robust metrics, we have to go one step back and we have to be able to reliably measure the system. And that is actually the challenge in a lot of biology, is are we sure we know what we're measuring?
So there will be a follow-up email that goes out probably later this afternoon or tomorrow morning with a link to today's event, so you don't need to take notes. You'll be able to look at the video again. Also, I'm going to include a link to an article that James gave me that was in Cancer Cell about how does a biologist fix a radio. And it's pretty funny for those of you who are into kind of sciencey, nerdy stuff. It's humorous. So just so you know, that is coming as well. So I promised folks that we're going to talk about time. This slide says human biology is dynamic. I want you to talk about that, but I also want to point out the line here, we don't yet know what we don't know, which is very key to what James is all about. But let's talk about the dynamic nature of human biology.
Absolutely. So all of us think of ourselves as the technical term is an N of 1. I think of myself, and sure, I might compare myself to population averages, but at the end of the day, I eat or not eat certain things or whatever, not necessarily because my doctor told me to, but because I've been living with myself for 35 years. I know what I'm about. And through that, we all develop an appreciation for ourselves of just how dynamic some of these different behaviors we have are. From when I sleep and exercise, how that influences my cognition the next day, all of this kind of stuff. From the biology perspective, we do know a lot about how dynamic biology is. I'd quickly piece together, just a handful of examples. When I think about studying biological dynamics, really the key point is to think about timescales. Biology operates on different timescales. We can think about things that are happening on the order of nanoseconds to seconds. That's a lot of the molecular interactions that happen. And when we start to kind of think about, physiological level behavior, we get different kinds of timescales. Things happen on the order of hours to days. Think about circadian rhythms. Can happen on the order of weeks to months. It's a lot of fertility cycles, menstrual cycle, months to years. There's seasonal changes that impact our biology. And what we're showing up here is that happens at the molecular level too. It matters when you measure these things at all of those different timescales. And to your point about we don't know what we don't know, there's some known unknowns here. There's some things that we know vary, but we may not have it fully characterized. But there's a lot of things that we just haven't really looked. How dynamic is it? Does it matter?
You and I have talked about cholesterol. What's your take on that? So just some examples of where time comes in.
Yeah, so what I think what I had told you before is, so when I go to, when any of us go to the doctor, we get a blood panel done, you get back, a few dozen things on a piece of paper. And if any of you are like me, I look at some of these and I say, my cholesterol's a little high. Did you just catch me on a bad day? Is my cholesterol really high? Or if I had come back this afternoon or tomorrow or next Tuesday, would it be different? For clinical labs, like what gets measured at the doctor's office, the answer is yes, we know that's why those are clinical biomarkers. For all of the other technologies that we study and research, for many of them, we don't know because nobody's ever looked.
You and I also have talked about vitamin D. There's a correlation there, sort of around time. What's that?
Yeah, that one's on a slightly longer time scale. That's more of a seasonal variation. Certainly it can be impacted by sunlight. We can absorb some amount of vitamin D in our skin, but for the most part, it also is seasonal just in the fact that we're outside exercising more in the summer. If the days are longer or shorter in winter and all of this, that it influences our behavior, which can have these trickle-down effects to our molecular biology as well.
And does it matter when somebody takes a vitamin D supplement?
Vitamin D specifically does need to be taken with food. It's fat soluble, not water soluble. I think it's A, D, E, and K are the vitamins. I don't know. Somebody's going to check me on that. But so that one does. But as to does it matter in the morning versus the evening? I think a lot of that's more of the cutting edge of research. Does it matter if I take my statin in the morning or the afternoon or the evening? It may matter. It may very well, but maybe we just haven't looked yet.
So let us talk about the time study. Give us the overview.
So talking about all of these dynamics, I've been trying to get this off the ground for about 8 years, and many thanks to Eric and many of the other wonderful people at Buck who are finally making it happen. The temporal investigation of multimodal elements. Pretty proud of that acronym. But the time study, the goal is really to define and map the biorhythms of what we call the human phenome. So we're all familiar with the genome. That's our genetic sequence that's largely static throughout our lives. The phenome refers to all of the other observable characteristics that are very dynamic. Glucose is part of the phenome, as is cholesterol. All of these different things, it kind of matters when we measure them. And so just taking a single snapshot in time is not particularly useful, either for research or for clinical practice. And so the point of this study is it's very discovery driven. We're going to enroll 150 people, we're calling them our time travelers, to embark with us on, it is an intense study. There's an 11-week main period, and then we'll have 6 and 12-month follow-ups. Enrollment criteria, we try to make it as broad as possible. For this first cohort, really the only limitation is over the age of 55. There were a few kind of practical reasons for that, a few scientific reasons. Everything is happening here at the Buck in the clinical research unit that Brianna Stubbs has helped build and direct with John Newman over the last several years. Allison is our one of our clinical project managers who's helping run all of the clinical operations for this whole study. Could not be doing it without them.
Great. So there's a note here about funding. I haven't got the question yet, but I'd be willing to bet that I'm going to get it, because wherever I go, people go, how's the funding at the Buck going considering the current situation? All right, because everybody knows the NIH is not in the best of shape. But what's the funding for this one, James?
Yes, so the Advanced Research Projects Agency for Health, ARPA-H, is a pretty new funding agency just within the last few years. People may be a little more familiar with DARPA, the Defense Advanced Research Projects Agency. ARPA-H, really the mission is to fund moonshot health projects. And so the Buck Institute very recently won an award of up to $52 million to study a variety of different things. There's 5 major themes as part of this award, and the time study is one of them. And I'm using the word award there very carefully. This is not a grant. We have been awarded a contract by the federal government. We have milestones that we have to meet. ARPA-H is within Health and Human Services. It's within HHS. And I think more recently, it's actually kind of moved under the umbrella of the NIH, the National Institutes of Health. Despite all of that, however, because of the structure of this contract, we are confident that we have the ability to run this 4-year program, of which time is actually just one piece of the larger $52 million.
Good news for us. That's great. Okay, so let us talk about what you're actually going to be collecting. What kind of data are you going to get from these folks?
Yeah, so I started out by talking with Allison and Bree. I just wanted to measure everything, and then we had to kind of, reel it back in. So we're measuring all of these different things you see on the screen, with the exception of a couple of them, multiple times. I think it's 43 time points over the, year. Most of it happens, 41 of them happens in 11 weeks, and then we have these follow-ups. So the whole genome sequence happens once. A couple of the clinical things, the functional over there on the side, only happen once. But the rest of these are happening at every single time point. And we're going to analyze all of these data together. And that's really where I believe one of the biggest innovations, and why this has the potential to be such a seminal study, is often we measure all of these in individual studies, and then we try to piece it together after the fact. As you might imagine, it's very expensive to run this all in the same group of people, but that's where the power is. It's measuring all of these different, all of the proteins in the blood, all of the RNA, the DNA, the metabolites, all of the lipids. We're doing a lot of microbiome sampling. It's not just stool microbiome. I know that the gut-brain axis is a popular thing to think about and hear about, but there's microbiota that live in other places on our body, in and on our body as well. And so being able to sample not just the gut microbiome, but the vaginal, oral, and skin microbiomes could be very telling for certain things. And then a lot of digital trackers allow us to help fill in the gaps between those time points so that we can get more consistent information and hopefully develop some digital biomarkers as well.
So when you and I chatted yesterday, you expressed the hope that what comes out of this is sort of a Rosetta Stone. And I went home last night and just made sure I knew what the Rosetta Stone was. It involved what, Greek, Egyptian, and it was...
Was it Latin?
I think. And it was used to bring hieroglyphics together. You could actually understand what these hieroglyphics mean. So foundational Rosetta Stone, that's really important, right?
Yeah, so to my point a minute ago about measuring all of this in the same person, there's a lot of different ways to measure each of these different biological signals. So just looking at proteomics, for example. So proteomics refers to measuring all of the proteins in a system. There's different ways to do that. There's different technologies. They're not necessarily compatible in the sense that if we use one technology to measure your proteins and a different one to measure mine, we can't necessarily just do a one-to-one comparison. It's not apples to apples. Therefore, it's really important that we measure a lot of that in the same sample so that we can use that to do the translation. And that's where kind of the Rosetta Stone metaphor comes in. Is this can be the Rosetta Stone for all of these different technologies, as long as we're measuring all of them in the same samples. And then the key is that allows us and the rest of the scientific community to start to compare existing data sets across each other and then bring them together and analyze and integrate them so that it doesn't matter which technology you use, we'll be able to translate it.
So this guy's taking this stuff seriously. You have an advisory board, right?
Mm-hmm.
So what kind of input have you gotten from your advisory board as you put this together?
Well, as Bree and I were, going through designing all of this, we certainly acknowledged the fact that there's a lot of things here, and we are not experts in a lot of them. So we have, I think, 4 different scientific advisors: Stanford, Harvard, UC San Diego, UC Davis, who are weighing on different aspects. One of our advisors is a circadian rhythms and sleep and women's health expert, and she had some really interesting comments and feedback for us about time zones, and because we hadn't really thought, it is an intensive period, people may not have the time to travel in a certain window of this, but she pointed out that if you travel more than 2 time zones away, there's a lot of research that I didn't know about that gets into how that can influence your circadian biology, and if people in our study did that, it could influence the results. And so, having that kind of insight from an expert on an area that I know very little about helped us design and capture just extra variability and things that we really wanted to be able to account for.
So we've talked about some of the things that are possible coming out of this based on the data that you're going to be collecting from folks. Let's talk about the skin microbiome. What might be what's a possibility with that?
Yeah, so a few years ago now, I did some consulting for a skincare company, and their goal was to do personalized skincare. So you could come in and say you were dealing with acne. The goal was we could sample, it's just a swab. We're not cutting skin out or anything. It's just a swab, like a Q-tip. You just do a little swab, and then we can sequence it, and you're profiling all of the bacteria that live on your skin. As it turns out, what bacteria are living on your skin and the relative abundances of one bacteria to another can influence the behavior of your skin. And so if you're putting a certain topical on, again, maybe it's for some kind of acne cream, maybe it's just makeup, and you want to be able to have it be most compatible with your skin so that it doesn't cause irritation or breakouts or rashes, whatever it is. That's just one potential application. There's probably a lot more that we just haven't really looked into that much that might be more linked to certain types of diseases, potentially some kind of skin cancers or anything. It's quite possible that the skin microbiome plays a bigger role in those kinds of health outcomes than we think.
So that's an example of knowing what we don't know, right? Mm-hmm. Exactly. Let's talk about, so you're going to also do vaginal microbiome. What are you hoping, what are the possibilities there?
One of the reasons that we are excited about sampling the vaginal microbiome is mostly because it's a very understudied area. A term we like to use in science is data desert for just an area of study where not very much is known or exists, and women's health is a major data desert. And so one of the goals in this study, and again, this is a very exploratory study, we have some hypotheses, but the goal here is really to generate the data that allows us to generate hypotheses so that we can do hypothesis-driven research. And I would argue, I am certainly not a women's health expert, but I would argue that we don't have the data in that specific area of study to do hypothesis-driven research because we don't have enough data to generate hypotheses.
Got it, right. And there are so many, we talked about endometriosis, right? There's no treatment for that.
It's been developing, and I think maybe there's some, I know there's some clinical trials looking at different blood tests, but up until even just a few years ago, the only diagnostic for that was exploratory surgery, which insurance doesn't cover, as you would imagine. So, being able to just collect more data, study it, leads to diagnostic tools, therapeutics, and this is kind of the first step. It may not be as exciting as, coming out with some intervention, but the foundation, we have to build the foundation until we can get to translation and clinical application.
What's the deal with retinal scanners? How did that get into the mix?
So some of you might be familiar with the UK Biobank. It's probably the world's biggest and most valuable biomedical resource that's out there. It's funded by the NHS in the UK. And one of the things that has been captured for a lot of that population of several 100,000 people is retinal scans. And there's a faculty here, Dr. Kapahi's lab, has looked in the past at a couple different aspects of biology related to things that you can get out of an image of a retina. And he's looked at it in lower level organisms like mice and flies. And he's found some really interesting trends with aging. And that there's an opportunity here for us to develop some sort of non-invasive diagnostic. What if just from an image of your retina, it's not even uncomfortable. You sit there, it takes like 2 minutes, you get your retinal scan. What if we could, diagnose some kind of disease, or even just give you a snapshot of your health and give you a score and say, hey, you're aging 87%, and just from a non-invasive image. That's the kind of thing we're hoping for, and it kind of goes to your, the idea of the Rosetta Stone. Because this other really big data set has that, we want to make ours as interoperable as possible.
So I'm going to put this slide up because I think it's really cool because it shows the time. I got a couple questions, and this prompts me. I should have said this earlier. James is not an MD, he's a PhD. And also, anything that we discuss here today, please talk to your doctor before you take any related action. So to wit, given circadian rhythms, is there a best time of day to schedule surgery?
From the research I've read, yes, it's in the morning. I'm not sure that it's so related to circadian rhythm of the individual. I think it has probably as much to do with the hospital staff and all of those kinds of things. But there is actually a lot of data that suggests it's not necessarily up to you, but if it is, morning is better.
Cholesterol, since statin activity is circadian stage dependent, are there most effective times? What do we know about statins? Do we know? And do you know anything? Are we going to?
I don't know. I'm sure somebody does. I will say that some of the research we've been doing shows, it's very complex. There's a lot of different influences here. Circadian biology may or may not come into play with some of the efficacy of some of these drugs, but with statins in particular, we've also found that the gut microbiome can play a role. And so if you have a particular profile of gut microbiota, one or a different statin may be more or less effective for you.
So I just want to repeat how pioneering this study is, and for anybody, we've recruited 3 folks so far. Anybody who's involved in this trial, it's such a gift to the research community to make yourselves available and to be part of this. Folks who are in the time trial, what are they going to get out of it? There's some financial remuneration and some other stuff, right? Let's talk about that.
Yeah, so as a participant in the study, you do get paid. I think it's $1,250 that's kind of paid out over the life of the study. You do get to keep all of the wearable devices, an Oura Ring Gen 4, a Whoop Band 5.0. We give you a Withings brand scale to take home. You'll wear a Dexcom Stello continuous glucose monitor at a few points during the study. You'll get all of that data. You get to keep the wearables afterward. There's a little bit of data that comes back from the clinical labs. All of those data get returned to you, and those are the kinds of things that would be actionable by your physician. It's more detailed than the standard blood panel you would do if you went in for a physical, and so you'll get all of that data back, and we would encourage the time travelers to communicate that kind of information to their physicians as well.
And how about data privacy? That's a big issue these days.
Absolutely. That's always yeah, it's something we take very seriously. First of all, off the top, I'll just say that at the Buck Institute, of course, we do not sell any of the data. We're a nonprofit. We use the data for research, but we do not try to monetize it or sell it. For the time study, I would say there's really 2 different angles here to think about for data security and privacy. One is kind of the anonymization of you just to the researchers. And so the clinical research staff will do the intake, and I've got some of their photos up here because they're the ones doing the real work. None of this would be possible without all of them. So they're the ones, these are the faces you see that help you walk through these different things. They're the ones taking your retinal imaging and helping take blood and all of this kind of stuff. They log all of the data and they de-identify identify it such that those of us who are doing the computational analysis never see any names or identifiable information, anything like that. On the back end, on the data security and data privacy side, IT here at Buck is really incredible, and they do some really great work to make sure that all of the data is stored very securely on servers located here at Buck and in the cloud, so that when we go to do our research and do all of the analysis, only those researchers who are supposed to have access, so that we're just making sure that we respect all of the data privacy and security reasons that, certainly I would be thinking about if I were enrolling in a study as well.
And all the samples are going to be frozen, right?
So that is yes, I hope that healthcare goes this way one day too, but especially in research, I made a comment earlier about being an N of one and thinking about yourselves. Comparing us to population averages gets us only so far. What's really useful is to compare me to me. And so if we take samples over time and we bank them, imagine a world where, let's say I get diagnosed with some disease 10 years from now. What if I had blood banked from today and next year, and I had this, this longitudinal trajectory of all of these samples, so that the doctors aren't just looking at this snapshot and then trying to treat me based on this one moment in time? What if they could actually go back and reanalyze samples in this new context of, hey, James developed this form of disease, this cancer, and then go back and look? Wouldn't that be extraordinary? That would really accelerate making data-driven clinical decisions.
So getting great questions. Somebody wants to know, how do we sign up for this study? So outside in the atrium after this event, we have folks from the clinical research unit who can answer your questions, get you signed up. Also, there in the email that you're going to get, there's going to be a link to the page on our website where you can sign up and show interest. So folks want to know, if you participate in the study, do you have to be available physically throughout the year? And somebody else wants to know about, can you travel? So let's get those details covered.
Yeah, so a lot of the IE criteria, once you talk to one of the CRU staff for screening, they can walk you through all of the enrollment criteria and everything. The traveling, it's only for that first 11 weeks, so we really ask you have to be within 2 time zones of Buck for those 11 weeks. You can travel the rest of the year, come back for your 6 and 12-month time points, that's totally fine.
Another question is, how are folks going to report changes in health, daily routines such as food eaten? So this is a really cool thing about this study.
Yeah, I didn't plant this question, but this is an awesome question.
It
lets me talk about, we're actually employing an AI chatbot that we developed here at Buck. We call it Bucky. You text Bucky. It's just a cell phone number. There's no app or anything to download. And the origin of this is a few years ago when all of the ChatGPT and the LLM craze was starting to happen. I got really intrigued by the idea of how could we use LLMs to help do my research, to help really just capture more data. And this idea of using AI to help data collection, that led us a few years later to Bucky. And so all time travelers have access to Bucky. It's just a cell phone number, and we encourage them to text Bucky whatever you want throughout the day. Sometimes Bucky will text you because you're going to be wearing a lot of wearables, and so if Bucky detects that you went for a run on your Whoop band, you might get a text from Bucky and just ask, hey, how was your run? On a scale of 1 to 5, was that really intense, not so intense? You can take a photo of whatever you're eating or drinking, send it to Bucky, get it logged, so that we can track and try to just incorporate all of these extra data points about everyday life. Because I will say, again, thinking like an engineer and not so much a scientist, one of the challenges we have with dealing with humans is free will and behavior and all of these kinds of things. I can't just put humans in a little cage and do everything. That's fine. So, when we're, for example, an easy one to think about is I have a lot of wearables on. So if I'm looking at the data from some of these wearables and I see a heart rate spike and maybe my blood pressure shifted, whatever it is, why did that happen? Is it because I'm up here talking in front of a bunch of people and I'm really nervous? Is it because I just worked out? Is it because my girlfriend broke up with me? Like, what happened that drove that physiological level response? We're using AI to help close that gap and just provide what we think of as ground truth labels. So what was happening throughout the day that links up to all of these things, so that whether it's wearable data or some of the signals that we might see in the blood, if we see stress spikes from cortisol or whatever else we're measuring in your blood, was that due to something that we're now aware of? Comes back to the known unknown kind of idea.
So folks want to know about the time commitment. So there are going to be specific days. You're going to spend a lot of time here at the Buck.
Yes, so this animation behind me, so this is detailing 4 days. Everybody comes in for what we're calling this high frequency period. It's a Thursday, Friday, Monday, Tuesday. So the larger study, you come in once a week for 11 weeks. These high frequency days replace 2 weeks of that 11, and you come in and you spend all day at Buck. The idea is that instead of just doing a once weekly blood draw, we're taking blood at 8 and 11 a.m., 2, 5, and 8 p.m. We're providing meals. You're wearing all of these wearables. There's a lot of questionnaires and different kinds of surveys. We're very excited to work with our partner BrainHQ that some of you may be familiar with. It's an app that you can download off the App Store. BrainHQ has designed their own cognitive assessment protocol to ask exactly the same questions that I'm asking in the time study, which is how does cognition and different aspects of cognition change throughout the day? They've designed this really cool set of assessments and in this neat study design that meshes with everything else happening in time. And so throughout these days, you're going to be doing these BrainHQ assessments as well. So these 4 days are very intense. You're here all day at Buck.
We have a lounge, just so you know.
We built a lounge
a really
specifically
great place
for this.
to hang out. Yes.
Yes, thank you to our patrons who's helped support us and built us a lounge. And then there's 2 other study period, 2 other challenges that we do throughout the Buck that take a little bit more time. But most of the other activities are, it's like, I think it's a couple hours, but again, I would ask the CRU staff.
So I think this is really cool. You have planned meals, right? Mm-hmm. Folks are going to get, this is what you're eating. Why are you doing that? Who's the partner? Why is this so cool?
Yeah, so I'm very sad to have seen him leave, but Noah Levine was on my team. He's a dietitian. He just left to go to graduate school, so I'm excited for him and sad for myself. But Noah, when we were designing the time study, I tasked him with, forget about budget, just design for me the coolest nutrition study you can think of. And he came back with this really neat study design that we then had to fit within budget constraints and, what the clinical staff, what Bree and Allison told me, yeah, people aren't going to do that. You can't do that. And so we are providing pre-prepared meals for people at different aspects throughout the study. It's not the full 11 weeks. It's just a handful of days throughout the study. And what we're doing with this is, I just told you how we're profiling everything about these people. One of our scientific advisors, Justin Siegel at UC Davis, he does all of that same kind of profiling on food. And so we're profiling these pre-prepared meals that we're working with a vendor to supply that have certain energy and nutrient requirements that have been mapped to each individual. It's been personalized to how much amount based on your resting metabolic rate that we're measuring. And then we're generating all of this omic data that I've been talking about on the food. You eat the food, and then we profile the blood. And so it's going to allow us to get at some of these really fascinating questions in nutrition research, which again, I'm not an expert there, but I have an advisor who is, that allows us to think about that aspect of health in the context of dynamics and all of these other things that we're looking at. So this is not a nutrition study, but we have built in this little piece to think about nutrition and to profile it so that as we have all of this other data, this could be an excellent pilot to launch some future precision nutrition studies.
I think you're going to analyze the food going in and going out and compare it to the food. That is, because you eat things and you go, is this really helping me? I mean, blueberries are good for you, but it's like, okay.
But are they good for me or are they good for you?
Exactly.
It might vary. And one of the interesting things, and I would just encourage people to Google, I didn't know this until I met Justin. There's an initiative, worldwide initiative called the Periodic Table of Food. Look it up. Really interesting website, and it's a nonprofit where all of these
I'll
researchers.
Include that in the email.
Yeah, all
okay.
these researchers contribute their, the data to the Periodic Table of Food initiative, and so that's part of what the Time Study will be joining and helping to supply some of the interesting aspects of precision nutrition.
That's great. Okay. A lot of people, lots of interest. This is exciting. So folks want to know when the study starts. It started.
It already started.
We
have 3 people enrolled and we have a waiting list already, so get in there.
And how long will it run?
Enrollment is going to be open for 2.5 years, starting from last month, 2 months ago now, June. And so, yeah, enrollment open 2.5 years, and then we'll be analyzing all of the data concurrently. As data comes in, especially the wearable data, we'll be getting that live, basically, and we're already starting to analyze some of those data.
So, really, really great questions. Do you have a hypothesis, expected results of the time trial? So we've sort of talked about this before, but how does this effort what does it say about the scientific process? Is there a hypothesis? What are you hoping, what's going to happen?
Yeah, I do have a few hypotheses, but largely speaking, this is a very discovery-oriented project. It's what we call hypothesis-free. So most of the research that is funded by the United States federal government is hypothesis-driven research, and it's very incremental in nature because that's you make a hypothesis, you test it, and then you look at the results, and then you generate the next hypothesis, and you go. With the advent of big data, and especially AI and the tools to analyze big data, we're able to start doing hypothesis-free research, where as much as I may try to not introduce bias into my thinking, if I generate a hypothesis, there is some level of bias there because I'm the one who came up with it and I have my own training and background and thought process. I do a lot of hypothesis-free research. I let the data speak for itself. Of course, there's caveats and there's challenges there, as you might imagine, but that allows us to take a more objective view of data and start top-down. And then we can merge that approach with the bottom-up, what we know about how things work, and then we can meet in the middle and hopefully figure out how they actually work.
Got it. Perturbing the system. So what is this about, and what are folks going to be doing who are enrolled in the time study?
You led this off by asking me about being an engineer. This is a key part of being an engineer. Just sitting here and watching something doesn't tell me very much about it. I need to poke it and see what happens, and then I can learn about it. And so we're going to do exactly the same. We're not going to be poking you, literally, but the pokes that we're doing, the 2 challenges, are the oral glucose tolerance test. This is a gold standard for diagnosing diabetes. It's a standard of care for pregnant women to look out for gestational diabetes. And so we'll be doing an OGTT. You come in on one of those weeks, you do your weekly time point, you take this glucose shake, and then we sample your blood at all of these time points after that. This is testing how your system responds to a bolus of food, your glucose insulin regulation system. The other challenge that we're doing is a cardiopulmonary exercise test, a little more colloquially known as a VO2 max test. VO2 max is one of the few metrics that really does correlate with longevity, and it's something you can train for. You can raise your VO2 max. I think it involves doing a lot of wind sprints. Doesn't sound fun, but it is modifiable. But it also tells us a lot about your system and how you respond to stress in the form of exercise. And so study participants will be doing both of these. And Lizzie and Joe are on staff at the CRU helping implement and run some of these for us. And so this is our way of learning how these things work. It's not enough to just take a blood sample. We need to take a blood sample, perturb the system, and then see how it behaves and how that change propagates throughout the system so that we can actually learn how the system works.
So this is what blows my mind. So you're going to get all this data. How do you make sense of it? I mean, it's like, I just think about, it's like such a huge amount of data. And how do you start? What do you do?
Well, I give it to these two, and they're the ones who actually know. Yeah, I mean, it's a great question. Right now we think about going to the doctor and measuring dozens of data points. I just told you we're measuring tens of thousands of data points at every blood draw, not to mention the continuous data that I get from all of these wearables. One strategy that we take is we map all of these different data onto a graph structure. We call this a knowledge graph because we can embed nuggets of knowledge in there, as well as actual numerical values for something. So if your fasting plasma glucose is 105, we can represent that in the graph. We can also represent that glucose is impacted by this genetic variation, that if you take metformin, there's a link between your glucose level and metformin, and all of these kinds of things can be embedded in the graph. And then we have a lot of really interesting mathematical tools, and this is a lot of what Amy Glenn in our group has been, she did her PhD in graph theory and computer science and looking at how we can traverse the graph and make different connections, because at the end of the day, if we just look at all of these data together, the more classical, traditional way of analyzing data is to just correlate things. We just plot things together and we see how well they match. Mapping them to a graph structure allows us to draw new connections that we wouldn't necessarily have made, because I don't necessarily know that two things are connected, but when we layer all of the information and we map it to this structure, that information is there, and then we can follow that connection, and we can even derive new edges, is one of the biggest challenges but opportunities in this kind of area.
So I want to talk about wearables. So wearables are a big deal in this study. I think you've talked about it. It's going to help you sort of bridge the gap, right? When folks are not here getting their blood drawn, you're going to get information that's going to help you correlate stuff, right?
Yeah, because if we think about how medical practice works today, it's very episodic. I go into my doctor when I go into my doctor. Maybe it's every year, maybe it's every 6 months, maybe you're young and invincible like me and you go every few years, not as much as I should. What happens in between those visits? I mean, that's a data desert on our own timelines. And so having continuously collected data, especially if we can do it in the home, remotely, allows us to bridge the gap between that very episodic nature of interactions with the health system. And for all of our time travelers, we're going to be analyzing all of the wearable data pretty continuously, put into these nice reports, and then you'll get this at the end of your 11 weeks, and again at 6 months and 12 months, as we go in and hopefully find some interesting relationships. And like you said, the goal here, a lot of us think of biomarkers as maybe some molecule that we might measure, and that helps us diagnose us for something. Digital biomarkers are also a really important thing to think about, and that's potentially a kind of almost sci-fi and in the future of, could we develop digital biomarkers for some of these things, not just having to do an invasive blood sample.
So you're into wearables.
Yeah, I saw you eyeing all my wearables right here.
So what are you doing there, James?
So everybody in the study is going to get an Oura ring and a Whoop band. I'm a nerd, that's okay, I'm not afraid to admit it. And a lot of what I've been curious about as we start to analyze these data, well, does it matter which finger I wear my Oura ring on? I teach a graduate level engineering class in Dallas about wearables and digital health. We buy the students Oura rings, and then I teach you about data science and AI and machine learning using the data that you collect on yourself using your Oura ring, instead of me giving you fake data or whatever off the internet. And the first thing my students always ask me when I give them the ring is, which finger do I wear it on? And in the past I said, that's a good question. Oura tells you to wear it on one of these two fingers, but I trust Oura, they're great friends, but I wanted to look for myself. So I've been, for the last 6 months or so, wearing multiple Oura rings, because I was curious, what is the inter-device variability? If I wear 2 Oura rings on the same finger, like I'm doing here, Do they give me the same answer? What about wearing it on a different finger? Is wearing it on my pinky? And what about on which hand? Does it matter which hand I wear it on? I'm an engineer, I gotta know. But that's the kind of thing that, it could influence what we learn from this. And I will say that the data's been super consistent. I would say it doesn't matter what finger you wear it on. Or if you're like me and you wear 4 of them, that's probably a little overkill.
So when we talked yesterday, you mentioned about really that hopefully this digital information is going to can be used now clinically. You used an example of your mom.
Yeah.
So this is for folks here because unfortunately not everybody's going to be able to be in the time trial, but everybody can use wearables. So let's talk about the value of wearables.
Yeah, so what I'm showing up here, that's not just something I drew up in Microsoft Paint or Figma. That's actually my Aura data. This is an auto-generated report that comes from everything that we collect off these rings. I had been talking to you, so my mom has been going through some health stuff recently, and she had a kind of acute episode of we weren't sure. But took her to the hospital, and she's had a string of doctor's appointments. I had bought my mom an Aura ring, and she's been wearing it. And so my brother and I, my brother is more into the AI space, not really biology. The two of us dove in, got her Aura data, generated these reports, and then I started looking at the data, and after we fired her PCP and got her a new concierge doc, I took it in, the data, and I actually have been texting back and forth with her new PCP about her Aura data, and then when she goes in for an appointment, they sit there together and they go through it. And it actually helped him treat her. One of the problems is that she wasn't sleeping, and so that helped him get to the bottom of it, but then after he started intervening, it allowed him to say, okay, great, now we can get you off this sleeping pill. Like, I don't want you to be on drugs if you don't need to be, so, I'm giving him all the data, and then we're going through it together, he's going through it with her, and so, kind of anecdotal, but that's not sci-fi. And there are medical practitioners out there that are very open-minded and will work with you on this kind of stuff. I will say the onus, though, is on us. It's on the scientists to make it interpretable. Because, I love her new PCP, but the guy doesn't have training in this. I have a PhD in this thing. It's hyper-specific. And so the onus is on all of us researchers to help bridge the gap and translate it so that I'm giving him what is most useful so that he can use it to help treat my mom.
Very cool. So James talked about his Aura ring. So he's going to talk about what he's discovered, and I hope that you take this away, that this is some of what you might discover should you get an Aura ring or some other sleep tracker, because it's important, right? Sleep is important. What did you find out, James?
Yeah, so I think one of the things that I was talking with you about was I've always thought of myself as a morning person. I like to go to bed early and get up early. I certainly did in grad school. More recently, though, I learned after looking at my Aura data, I thought I went to bed by 10 p.m. every night, but I definitely didn't fall asleep at 10. And so this circular thing behind me that's kind of looking at a clock, the blue line starts when I fell asleep, and then it ends when I woke up. And the histogram right next to it is just showing what that distribution looks like. So why is that important? Well, turns out that sleep consistency, and I'm not even talking about timing, I'm talking about timing in the context of when I go to bed and when I wake up, is really important for health. I've talked with Dr. John Newman on faculty here at Buck. He's an MD, a practicing geriatrician. Talked with him a lot about how he thinks about interpreting some of these data in the context of treating his patients. And things like sleep efficiency. That's the percentage of time that you're in bed but awake. How that changes. We all get worse at sleep efficiency as we get older, but that's actually a really important thing for him to know and be aware of when he's trying to treat somebody. If any of you have sleep trackers, it'll calculate a sleep score and this kind of stuff. And so that's what I'm plotting up there at the top is just my sleep score. Okay, so that's some kind of aggregated score or whatever. I was looking at that and I was noticing all these huge dips and I was like, what happened during some of those? And then when I went back and I looked at my calendar, I was able to annotate the graph and show, Turns out I don't sleep so well when I travel, no surprise. But like when I got COVID a couple times, yeah, my sleep kind of cratered. And even after I was no longer symptomatic, it took like 15 days for my sleep to regress back to the mean. And that was interesting for me to understand that even though I didn't feel sick anymore, clearly I still needed to be giving my body the opportunity to recharge. Now, I labeled those manually, but here's the cool part. This is what Bucky's going to do for us. This is what AI is going to be doing, and we're doing it right now. I showed Emma's photo, Emma Glass is in my lab here at Buck. This is the kind of stuff we're working on right now. I'm not going to be manually labeling this stuff in the future. You're going to be texting Bucky, hey, I worked out, I took a nap, I ate an apple right before bed, I ate chocolate, whatever it was. And then Bucky's going to analyze all of this for us and give us, interpretations and identify these trends. That's not science fiction. I mean, that'll be here within the next few months. We're going to be rolling this out to our time travelers.
Very cool. So this is a good question. Is there a data feedback loop? Do you expect behavior changes, lifestyle, diet by participants? So that may come in the next trial, right?
Well, that's a very interesting question because it's one we asked ourselves many times when we were designing this. It's one of the challenging things about studying humans is we might perturb the system. There's kind of a Heisenberg uncertainty principle here if you're more into like quantum mechanics and quantum physics. The idea of by observing a system, I've perturbed the system, and so I've changed its behavior. We actually had a lot of conversations about should we blind all of the wearable apps? Because if you've never worn a wearable tracker before, and now you're seeing the data, your behavior is probably going to change. How does that influence everything that we're trying to study and measure? We talked about that a lot, and basically what we landed on is we know that we can't capture all of that variability, and so we're basically not going to try. We just want to observe people doing whatever it is you're doing. My mantra as more of a data guy is as long as I've captured the information, as long as I have that data label, and that's where Bucky is so important, as long as I have that data label, then I think we can work it out and understand the influence of some of those things.
So this is going to help us wrap it up. What's the biggest bottleneck to personalized preventative care? Is it lack of long-term longitudinal data, sensors that aren't good enough, models that aren't good enough, we don't understand the biology? Like from your perspective, what bottlenecks are you hoping to be able to?
Well, those were 4 really good ones, and the answer is yes, 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 and yes. I would say, there's, where do we start? I think one of the biggest problems I see is data silos. Data are really siloed, meaning they're separate, they're independent, they're not kept together. If I just think about my own health record, for example, my own medical chart, I'm not that old. I was in school not that long ago. I've lived in 5 different states in the last 10 years. I have no idea where my medical record is. And even if I wanted to get it, I couldn't because I've interacted with so many different health. It's very siloed. So that's one challenge. I think AI is going to really help us bridge some of those different silos together and just lower the bar, lower the activation energy that's needed in order to kind of merge those together. A lot of the sensors and the technology, there's a lot of people working on that. There's a lot of cool stuff happening at Buck on technology development. But I think a lot of it is also just some of the interdisciplinary stuff. It's where you started. It's why a place like the Buck is so extraordinary, is because even though everybody in this institute is studying aging, we're all studying different pieces of it. And there's actually maybe less overlap than you would think in all of the different areas of expertise. But when we bring all of these people together and we're starting to bring more translational and clinical focus, we're starting to bring more computational and AI focus, I think that's where some of the biggest opportunity for all this lies, is just integrating all of that knowledge. It's synthesizing all of this stuff together till we can get to the point where we're really deriving and driving new insights and discovery.
I also want to point out that all the data and all the samples that are being collected, they might be engaged with other Buck labs, right?
Absolutely. Yeah, I mean, I only mentioned a couple different labs here, but the entire institute is going to be working on this. I'm probably going to be analyzing the data from the time study for the rest of my life. I mean, there's so much here. It's so exciting. I've been trying to do this for 8 years.
Thank you, James. Pretty cool stuff, huh? So, I got a lot of questions specifically about the trial, who's eligible, what's involved, how long can you travel. Folks out in the lobby are going to be able to answer those questions for you, so please. So thank you, James. This has been a hoot. Our next community seminar is not to be missed. It happens on September 3rd. James mentioned Dr. John Newman. He is on the Buck faculty. He's also a practicing geriatrician at the VA Center in San Francisco. How to be your own geriatrician. Do not miss it. We have a critical shortage of geriatricians in this country, and John's going to have some really good advice to help you get the best care possible. So September 3rd. We've been talking about the time trial. We have other clinical studies happening here at the Buck. You can get information out in the atrium, talk to folks. It's also on our website. You'll get a link to it. You can learn more about what we do and become a smarter person, by enrolling in our Buck Academy. We have a great online class that's all about the biology of aging. It's got great reviews. We also have launched the new season of our very popular podcast, We're Not Getting Any Younger Yet. So we interview researchers from around the globe, so that will be interesting as well. If you have not been on a tour or haven't been on a tour in a while, please sign up. They happen twice a week. We now have a museum exhibit as part of our tour that sort of goes over the history of the Buck and the history of research on aging. Also keep up with us. We're out there on social media. You can go on our website. You can sign up for our newsletter. That's where you really get all the information. And also, just so you know, we have opened registration for our September seminar. So it's going to be popular. Sign up, get on the list early. Thank you so much for coming. It's just been a really, really great experience. Enjoy your summer. See you in September, everybody.
Thank you.